I'm very pleased to introduce our speaker today, our very own Paul Fine, a professor in integrative biology. Research in Paul's lab, he says, investigates the origin and maintenance of tropical tree diversity using a combination of field experiments and molecular phylogenetics. So Paul, here's a fun fact. His first scientific paper, which was in 1999, is an MVZ publication entitled Aerial Predator Recognition by Free-Ranging Scaloporus Occidentalis. That's funny, I didn't know they were trees. Well, he clearly went over to the dark side after starting work on vertebrates, and for that we'll never forgive him, but I'm sure what he's doing, and he'll tell us about today, is very interesting. Paul was an undergraduate here at Berkeley. He got his PhD at the University of Utah, and then was a postdoc at the University of Michigan before coming back as a faculty member here. This is Paul said that as an undergraduate he took MVZ lunch and he sat in the back, and but this is your first time as an MVZ lunch speaker, and so we're very happy to have you start the semester for us. Thank you, Paul.
Thanks, Michael. Yeah, it's a real treat to be here in this room. I still remember when I was a senior in undergrad, sitting probably right about here, and it used to go that way, and there were all kinds of interesting professors in here that would be Professor Stebbins, Professor Patelka. They were alternating sleeping and arguing sometimes both at the same time. And I just remember being blown away by the presentations here. It's a real treat to be here. So the title of my talk is Ecological and Evolutionary Drivers of Geographic Variation and Species Diversity, which is the same title of a review paper that I wrote last year that was published a month ago. And we have about 45 minutes, and I'm a really fast reader, so I just thought I would... I apologize for those of you who saw me make that same joke last week, but
it still works, Paul.
I know. So, you know, like a lot of people in ecology and evolutionary biology, the latitudinal gradient in biological diversity really drew me in to this field. And my first experiences in the tropical rainforest, where incidentally you can find as many trees in this photograph as you can in the entire United States and Canada. I was just completely mystified by the tropical diversity. And you know, just to give you some figures in trees, we don't know how many species are in the neotropics, somewhere between probably 20 and 25,000 species of trees. And as I said, there are only 600 species of trees in the United States and Canada. So it's a really big difference, and the very same kinds of dramatic differences in species diversity in vertebrates as well, the birds and amphibians and reptiles and mammals. And when people speak about the latitudinal gradient, and as they've done so over the last couple centuries, they often say and it's unexplained, and so that also that element of mystery really drew me in as an undergraduate, and I really been focused on that question ever since I started studying ecology and evolutionary biology. And I think part of the reason that people have kind of punted on the latitudinal diversity gradient is because when thinking about why there's so many species in the tropics, tropical rainforests have a lot of these characteristics. They're warm and wet. They're productive. There is a lot of energy flowing through tropical rainforests. They cover a large area. They're climatically generally stable compared to other parts of the world. They're ancient. There's a long evolutionary history, a fossil record of tropical rainforest biota. And they have complicated food webs. And so they have all these things all at once. And so trying to disentangle which of these factors, which are at the center of many hypotheses to explain the latitudinal diversity gradient, is difficult, because all these things are happening at the same time. And really, my aim today is to try to review some current understandings of in ecological and evolutionary theory, and sort of some of the advances that we've had in the last couple decades, to maybe think about how we can disentangle the relative importance of these factors. And I think today is a pretty exciting and scary time to be a diversity scientist. It's exciting because we have so much data at our disposal today, than, especially compared to just a couple of decades ago, in terms of the reconstruction of the tree of life. We know from many major groups of organisms, at least definitely the vertebrates, but also lots of the higher plants, many clades of invertebrates, we have very detailed reconstructions of the tree of life, including fossil calibrations. So we know now approximately when major clades of organisms evolved, and how many species there are, and how they're related to one another. And that's a really major advance that 50 years ago, ecologists and evolutionary biologists couldn't draw upon that information. We also know a lot about the geographic distribution of organisms. There, especially for vertebrates, we have really fine-scaled maps for the entire globe of where every species lives. And it's also a scary time because half the world is being used by agricultural and industrial production, and we also have this year, 2015 was by far the hottest year on record, and it's not really under dispute that the world is the climate is changing in the world. And so when you think about the evolutionary and ecological drivers of diversity gradients, land area and temperature are right at the center of the theories to explain diversity. So I think it's really important that we think about and understand how these factors influence diversity gradients if we want to make some good policy decisions in the future. So today what I'm going to do is talk about diversity gradients and talk about differences in diversity between different parts of the world by really focusing on the regional scale. And so I'm going to talk a lot about latitudinal diversity gradients, but really what I'm talking about are differences of numbers of species between different regions. And that applies to the latitudinal diversity gradient, and most people that talk about diversity, they often refer to this gradient, but I just want to emphasize that you can make any comparison you wish. It doesn't have to be in a latitudinal way. So I want to start at the regional scale, and the reason I want to do that is because I want to think about how species evolve and how they and how those populations are kind of don't go extinct. And a reasonable starting point is to think about biomes, because biomes are they kind of form boundaries where you usually don't get species crossing them, and so these biomes can be thought of as really like evolutionary arenas. And why is that? Why do you not find species crossing biome boundaries? And definitely there are exceptions. There are things like mountain lions that live everywhere. But if you think about most major clades of organisms, you find in different biomes they have completely different species compositions. And that's because, for a number of reasons. One might be that the organism never arrived, say that it never dispersed and got to an adjacent biome. And I'm not going to talk too much about that. I think that's not a very interesting explanation. And given enough evolutionary time, most organisms are able to disperse most places in the world. So what I'm really going to focus on is that organisms can't just survive in different climates. So biomes are generally mapped. The definition of a biome is the climatic differences. And so, and a very simple example of this is, for example, frost. Organisms that don't have frost tolerance, if they disperse to a biome where it freezes, they're going to die. So that's one explanation of why you get turnover in species composition across biome boundaries. Another one is that they could survive, perhaps, but it's difficult to maintain species cohesion across such a strong climatic gradient. And that's because many organisms have evolutionary trade-offs where you might have an adaptation for one kind of survival in one kind of climate, but that comes at a cost. And back to the frost tolerance example, organisms that invest a lot in frost tolerance for the sugars and alcohols in their leaves or going dormant, that's a use of energy that could be used for competing in areas where there is no frost. And so if you compare a frost-tolerant tree versus one that is not frost-tolerant, you find that the tropical tree grows much faster. And the implication is that it's much more competitive in frost-free environments. So I'm going to suggest that these kinds of trade-offs underlie the turnover in species composition across lots of biomes, not just between tropical and subtropical, but say across dry and wet gradients as well. And so when you think about these biomes around the world, and this is from a paper that I did with Rick Ree about 10 years ago, you have to acknowledge that these biomes have changed through space and time. If you think about the tropical neotropics, for example, today, when you go back to the mid-Holocene or the last glacial maximum, and then to the Miocene, the Oligocene, and the Eocene about 55 million years ago, you'll see there's been a major change in the area of the tropical rainforest. And in this world, in the Eocene world, most of the world was tropical rainforest, or at least tropical, never freezing, and there was no ice at the poles. There were temperate regions where there was freezing temperatures up here, but no permanent glaciers at the poles. Those come about much later on in time. And so if you think about, say, boreal regions have less evolutionary history. They used to cover a very small area, now they cover a very large area. And that's what I'm thinking about when I'm thinking about biomes through time. And so what we did in this paper was just map these biome areas through time, and we divided the world into these 11 temperate, boreal, and tropical regions. And I'm just going to take a few minutes just to go through this. So this is basically just time versus area. Area is on this axis, and here's time. So these are the tropical areas, and just follow one of the colors. The different colors just correspond to different interpretations from different people who made paleo maps. But you can see that a very large area here in the tropics, it used to be bigger, and during the entire time though, there's been a very large area, say in the Asian tropics. So that what I call the time integrated area. And if you think about it, in every slice here, every slice of time, you have all the species that are living there, and all the populations, all the individuals that are in those populations, kind of cumulatively going through time, living in these large areas. Whereas you compare it to, say, the boreal forest has a much more recent history. It used to cover a small area, now it covers a very large area. And so if you think about the cumulative time area in those biomes, it's much smaller, say, for a boreal forest than it is for the tropical rainforest over time. And together with my colleague Walter Yetz, who's visiting actually Berkeley right this semester, and Stanford, we kind of extended that thinking to not just use boreal, temperate, and tropical rainforest biomes, but think about more major biomes in the world, like grasslands and tundra, Mediterranean, woody savannas. And we did the exact same exercise where we mapped these through time using what we could find out about the history of those biomes. And then we added an additional variable, and that's this red stuff, and that's energy, with the acknowledgement that with more resources, you could have larger population sizes in those areas. And so you have time integrated area with energy. But it's the same sort of idea of that these different biomes, and I'm going to use this term biome and bioregion interchangeably, have different sizes, they have different evolutionary histories, and they have different amounts of available energy. And so you have these different, basically, evolutionary arenas through time that produce differences in species diversity. And a really critical justification for this approach is actually looking at the data. And here's our looking at birds, mammals, amphibians, and reptiles, and calculating what is the overlap in species composition across these different bioregions. And if we had a really high overlap in species composition on average, then then those maps that we produce really don't have much towards reality, because then that would mean that those biomes really aren't acting as evolutionary arenas. But if you have very low species composition, then I think that this approach is, provides critical justification to use this kind of scale as thinking about what are the evolutionary arenas, where did this biota come from, and how have they persisted through time. And we found very, very low species overlap. And birds, as good flyers, were the only exception. So it's probably we should have redrawn the maps a little bit for birds, but anyway, we can come back to that. All right, so that whole thing was to emphasize to you that the regional approach I want to take. When I think about diversity differences around the world, I want to think about it at this really large scale, at this bioregional scale. And now, thinking about that scale, can we apply the hypotheses that have been offered for, say, the latitudinal diversity gradient, and think about how we may be able to disentangle some of those factors to explain diversity gradients. So I really drew on these 4 papers to go through my list of hypotheses. And Eric Pianka's 1966 paper kind of synthesized a lot of the existing hypotheses of the time. Rhodey 92 and Oikos, there was an annual review in 2003 by Michael Willig, and then Gary Middleback et al. In 2007 had a review in the Ecology Letters on evolutionary hypotheses to explain the latitudinal gradient. And really, what I want to do here is take those hypotheses, see where we can find some common ground, and think about how they influence speciation and extinction probabilities at the regional scale. And so there are about 20 hypotheses in those papers. And here, I listed many of them, and I put some of them together because a lot of these hypotheses were actually sort of the same thing. And then I further merged them into 5, basically. So I took these 20 hypotheses. 3 of them had already been rejected in previous reviews, so I was able to get rid of those. And so there's hypotheses about the large area of the tropics, this tropical conservative hypothesis that I'm going to talk about in a second, out of the tropics, and this available energy, so basically time-integrated area hypothesis that I just told you about. All those are merged here into this time-integrated hypothesis. Climate stability is a big hypothesis. We're going to talk about evolutionary speed and biotic interactions. I'll briefly talk about ecological limits. And so, thinking about these hypotheses at the regional scale, here if you get into my mind a little bit, what I'm thinking about is how do these different factors influence the speciation and extinction at a large scale. So these little circles are just, say, individuals, and each individual has its own genetic diversity, which could be high or low. These individuals are distributed around a geographic range, which is this axis, and then this moves through time. And if those individuals run into a geographic barrier, there's some certain probability that speciation is going to occur. Okay, and if it does occur, and that's showed by them changing into different colors, they may come back into sympatry, and they may fuse back into one species again, or maybe reproductive isolation has evolved, and then they're going to have to persist. So all of these hypotheses, they usually talk about one aspect, or sometimes multiple, whether or not a population is going to run into a barrier. What's the probability that a population is going to come into contact with a barrier? What's the probability that reproductive isolation is going to evolve? And then what's the probability that those lineages are going to persist? And the second way to think about it, so that's allopatric speciation, basically, very very simple classic allopatric speciation model. You can think about it also with this parapatric speciation model, where you have, say, 2 soil types or something, or 2 kinds of habitats, or this could even be a trophic resource. This could be, an insect feeding on a type of plant or something. And you have this probability that they're going to encounter the new habitat, probability that divergent selection is going to lead to reproductive isolation, and then again, the probability that these isolates are going to persist. Okay, so I'm going to, that's the framework that I want to think about. I want to think about how populations, through population size, how through geographic range and genetic diversity are going to influence the probabilities of speciation and extinction. So to start off, this hypothesis of time-integrated area and energy, this is the same framework. Thinking about population size, how is that influencing speciation and extinction? Well, large populations have large geographic ranges, and large geographic ranges may encounter new barriers. They may encounter new habitats in a bioregion. with enough environmental heterogeneity. Large populations also have high genetic diversity, and this is going to reduce extinction probabilities because of lack of inbreeding depression. Having a large geographic range also reduces kind of stochastic or catastrophic extinction that might happen if you have a very small geographic range size. If you have high genetic diversity, you probably have a high phenotypic variability as well. And high phenotypic variability is associated with being able to utilize heterogeneous habitats or trophic resources, okay? And that may lead to habitat or trophic specialization, which would increase speciation rates, but it also can reduce extinction probabilities in climate change. So if in these populations are living in a place where there's glaciation, if you have a very specialized population, say for a certain soil type, or excuse me, if they have very low phenotypic variability and the climate changes, then that's going to increase speciation. I'm sorry. It's going to increase extinction risk for those populations. Now it confuses everybody in the room. Okay. All right. So that's the way I'm thinking about it. And I want to separate out, okay, so back to population size again. This is, again, areas that have a large time-integrated area. There's a large area that have been persistent for long periods of time are going to have large population sizes. And I also told you that with more resources, you could probably pack more individuals into that bioregion. And that is different, though, than this other hypothesis of ecological limits, which was the subject of a very interesting debate 2 years ago at the American Naturalist Symposium about whether or not you had ecological limits at these large scales. And I'm going to punt on this one and say that I think this ecological limits hypothesis is really a distraction, because the idea is that bioregions with more resources could fit more species because they have, say, a larger carrying capacity, fit more species in areas that have more resources. But it's missing a really critical logical link there. It's not, the resources have to do with how many individuals you can fit in a bioregion, not how many species you can have. Think about it this way. It only makes sense if every species only has one individual in it. You could have, if you had 1,000 species and one of them had, say, 700 individuals, and you added another individual in, who's to say that that individual is not going to displace one of the common species? And so you could fit another species in there. You can always fit another species in. And so that's one of the things that I think is really missing from this ecological limits debate. And so that's why we're not going to talk about it. And I'm going to say that I think it's a lot more productive, a lot easier just to think about how energy influences probabilities of speciation and extinction at large scales. All right. So related to time-integrated area is this tropical conservatism hypothesis. And actually, the more I read about it over and over again, the more I was convinced that it's actually the exact same hypothesis. Because the idea of this hypothesis is that you have lineages, because of the large extent and age of the tropical rainforests, are more likely to have arisen in the tropics. And then because of what we were just talking about, these trade-offs to extend out to other biomes, those things are infrequent and don't happen very often. So those two observations together are used to explain why it is that you find so many lineages with tropical origins and why you get so few transitions to non-tropical areas. This is a hypothesis from Weems and Donahue in 2004. So lots of people have taken that hypothesis and applied it to phylogeny. So I'm just going to burn through a couple of just pick figures like this, which are totally incomprehensible. But these are just lots of species, and red is tropical, I guess, and blue is temperate. So you see, again, many lineages of tropical, transitions to the temperate zone are few. Species appear to accumulate in these tropical regions. The out of the tropics hypothesis by Jablonski is basically the exact same thing as the tropical conservative hypothesis and the time-integrated hypothesis. It's the idea that these lineages are originating in the tropics and they're sometimes dispersing out of the tropics. And this is using the entire mammal phylogeny, looking at speciation and extinction rates, using the molecular phylogeny to predict whether or not you get the same patterns. And, since this paper came out, Dan Roboski kind of trashed the entire methods for how they estimated extinction rate, speciation rate, so we're not going to go into it, but I'm just pointing out that you get these same sorts of patterns of lineages accumulating in tropical regions. This was a paper by Pyron and Weens on amphibians. Again, the same pattern of lots of tropical lineages, infrequent dispersal out of the tropics, and again, probably higher extinction at high latitudes and low extinction in the tropics. This is Walter Yates et al. In Nature on the bird phylogeny, again, tropical lineages. He actually didn't find any differences in diversification across with latitude, but he at the same time found that there was more accumulation of tropical lineages in birds, consistent with his tropical conservatism hypothesis. So almost every large-scale clade that has looked at this hypothesis has found support for it. Generally, lineages are from the tropics, and bioshifts are relatively uncommon. Okay, what about this direct test of this area energy time evidence? Well, that paper I talked to you about with Rick Ree, 2006, we looked at time integrated area and it correlated really well with tree diversity. Walter Yates and I used the global vertebrate database to correlate those 32 bioregions around the world with time integrated area and energy, and we found very strong correlations. Explained, what was it, 80% of the variability in biodiversity among bioregions. So we were pleased with that. Going on, Walter, working with his Bellmaker, correlated bird and mammal richness using those new data sets and looked at whether or not diversification rates also could play a role in understanding these differences among bioregions. He compared that with time integrated area, energy, and then temperature alone, and he found time integrated area was by far the strongest predictor of diversity. So I think there hasn't been that many tests, but I think this approach of looking at really large scales and thinking about time integrated area and the probabilities of extinction and speciation is one way forward to understanding large scale gradients of diversity. Okay, next hypothesis is climatic stability. And this one has been written about quite a lot and at a lot of different time scales. Jansen in '67 wrote about why mountain passes were higher in the tropics, and he was talking about how the variation within a year at, say, tropical latitudes was much smaller than at higher latitudes, and that may cause serious differences in physiological tolerances for the organisms and would limit their ability to disperse. Stevens kind of applied this to the latitudinal gradient and thinking about how that might influence range size at really large scales. And Denisius and Jansen in this paper in 2000 extended it to basically eras and thinking about how higher latitudes are always subject to climate instability. And here's a graph of the last 300,000 years showing the difference in climate variability in the tropics versus at high latitudes. You can see it's substantial. And that together with this evidence of smaller range sizes at the tropics compared to large latitudes, they put those two things together and they had this framework where large climate variability causes increase in species ranges, and that's going to influence specialization, range size, and probabilities of speciation and extinction. Does this sound familiar at all? Well, it's the exact same framework that I've just been hammering into your brain, or attempting to, and really focusing on range size and how it influences speciation and extinction rates and the probability of specialization. Whether or not you're going to have, in a climatically stable area, are organisms going to have then a sufficient time, and they just come up with this figure that organisms need 100,000 years. And, I'm not sure what the basis is for that, but the implication is you need some amount of time to be able to specialize on a habitat or a trophic resource. And so an example I'm thinking of is maybe a serpentine soil, or a particular trophic resource that it's going to take time for that sort of specialization to evolve. And in areas where you have glaciation happening and the climate's changing every few 10,000 years or so, that kind of specialization is going to be difficult. And if you are specialized, then it's going to actually increase your speciation risk at high latitudes, but not at low latitudes. And if you think about the same framework of populations size and area, here's a climatically stable area and here's a climatically unstable area. And if you say that there must be some minimum time for speciation to arise, well, in a climatically stable area, maybe this specialization perhaps is inevitable over time, and in a stable area. But if it takes time for specialization to evolve, then every time that there's a climate oscillation, that's going to knock down one of those newly specializing lineages. And so, what I'm thinking here is that time-integrated area surface that I presented to you is perhaps a little bit too simplistic, and that is because it might be these minima that are really more important than the actual time-integrated area over all time. It might be that extinction is really critical, and so the time-integrated area, this large area here might not really matter that much if you're thinking about speciation and extinction in this region. And so perhaps we should actually incorporate this kind of this idea into trying to calculate bioregional area and potential speciation and extinction. Okay, so what's some of the evidence for climatic stability hypothesis? Well, in general, what we know, extinction rates seem to be very high latitudes, especially for organisms that are poor dispersers. Some of the genetic diversity studies looking at Paleorefugia in Australia and in the Atlantic forest of Brazil have found that you generally find high genetic diversity in areas that are paleoclimatically more stable. And the few people that have tried to take measures of, say, climate oscillation, this guy Jansen again, and correlate it with endemism in trees and invertebrates, have found strong correlations. One of the most convincing studies to me was Jim Valentine's work with David Jablonski. And this is a paper that came out last year, and I kind of just stole from it. And that is that what they're talking about is diversity gradients in the sea. And the thing that's really cool about diversity gradients in the sea is that you get completely different patterns on land in some cases. And that's because in warm waters that are climatically stable, they can often be really not very productive at all. And that's because of the relationship between oxygen and productivity in the oceans. And so you can get, for marine invertebrates in tropical shallow seas, you have high temperature but low productivity. You get high stability of the water column, though, over long periods of time. You get high species diversity. Okay? So these are the temperate examples. Everybody's already familiar with this. You get where you have this, it's really difficult to disentangle temperature, productivity, and climate stability because they all kind of go together. Now, what's really even crazier is that these deep sea marine invertebrates, these are like way down low. They're so deep that you need to have one of those crazy submarines that have like walls thicker than this room, right? And they go down there, and every time they have a little scoop, a little robotic arm, and they scoop up some kind of a marine invertebrate. I don't even know what marine invertebrates look like. But they scoop them up, and every time they do, they get a new species, basically. The accumulation curves are out of this world. Well, these are high species diversity communities in crazy low temperatures. It's always 4 degrees, or I think it's 4 degrees. It might be negative 4 degrees. And no productivity at all, really. It's just like every now and then a little bit of plankton or something will float down to those depths at the center of the Earth. So here you have a situation where you have really, really high species diversity, low temperature, extremely low productivity, and really high species diversity. So if you think, what's the commonality between trees and vertebrates on land and marine invertebrates in the ocean? The only thing that correlates with it is climate stability, not temperature and productivity. temperature and productivity seem to be decoupled, you see, from species diversity. Okay, how are we doing here? Oh, terrible. Evolutionary speed. This was a hypothesis that was really championed by Klaus Rohde, in his '92 review. And the idea is that high temperatures elevate mutation rates in ectotherms and in plants. And this could increase the speed of reproductive isolation, thus the probability of speciation. It could also increase the speed of selection and adaptation, and specialization, and then by consequence speciation. So not only will it elevate mutation rates, it elevates metabolic rates as well. And this could increase the speed of generation time. If you have faster generation times, that can also interact with the speed of selection, increasing speciation rates. So unlike those other hypotheses I've been presenting, this one is really just focused on this one. It's focused on genetic diversity, and it's really focused on the probability of speciation occurring through reproductive isolation. Okay? So it really says nothing about extinction. It says nothing about the probability of running into a barrier. It's only at that phase. Well, Coyne and Orr in their book on speciation, they say, well, it seems that reproductive isolation is not generally limited by mutation rates. And you know, that's arguable. But to me, the real difficulty with this hypothesis is that how do you attribute genetic diversity to mutations from temperature? Because I just spent, you know, the last 48 minutes telling you that genetic diversity is also associated with population size. So larger populations are going to have larger genetic diversity. So somehow you need to be able to decouple things that promote genetic diversity, just normal garden variety things like population size and adaptation to different environments and environmental heterogeneity that may influence genetic diversity, from this additional effect of temperature increasing genetic diversity through mutation rates and through metabolic rates. So I'm not willing to just completely toss this hypothesis out because I think it's interesting, but I think that the way to really get at it is probably through experimental studies, where you can try to control some of these variables and see what happens. And you know, a lot of the hypotheses that are the papers that have been criticizing this hypothesis are looking at like phylogenies of mammals or birds. I mean, it was never meant to be to explain endotherms, except for indirectly. So I think that, you know, maybe it would be worth doing some really careful experimental studies, especially through generation time. I think generation time is something that really not many people have considered or studied in great detail. Okay, what about biotic interactions? Biotic interactions have been consistently on those list of hypotheses to explain the latitudinal gradient, and there's been a subject of several reviews over the last couple of years trying to quantify biotic interactions across latitudes. Generally they say that yes, there's more biotic interactions across in the tropics than in the temperate zone. So I'm trying to think how would this influence speciation and extinction rates, and I guess the idea would be when you have more trophic interactions, probably would be related to resources. They could affect speciation either through the interactions of biotic interactions with habitat heterogeneity, or through this geographic mosaic and reciprocal selection idea. And I'll tell you a little more about this in more detail. Or they could affect extinction rates through competition, perhaps through narrower niches, meaning that species are going to be kind of more stably coexisting because they're not competing over the same resources, or through another kind of offshoot of the narrow niches is this idea of enemy-free space, where organisms have maybe a particular defense where they are not getting hit by predators. And that may lead to a rare species advantage, which would reduce extinction rates in the community. So for speciation, I think maybe the simplest hypothesis is just that if their biotic interactions influence specialization across resource gradients, then you could have higher speciation rates in the tropics across environmental gradients. And let me just quickly, this is some of my own work studying habitat specialization by plants on different soil types, in high and low resource soil types. These are white sand forests surrounded by higher resource soils where you get often completely different plant communities on the different soil types, but they're often really closely related to one another. You have congeners that are specialists on different soils. This is one group that we studied in a group of trees where you have repeated evolution of white sand versus clay specialization. And one thing that we did was we've done reciprocal transplant experiments to see whether they grow faster or slower in the different habitat types. We've also looked at what are their natural enemies and how much do they invest in defenses. And we find good evidence for these evolutionary trade-offs where low resource soils have slow growth rates, high defense investment, and completely different complements of enemies than species that, even their sister species on a higher resource soil where they have faster growth rates, less defense investment in it. So the idea being that with more enemies, that these kinds of specialization to habitats that differ in resource availability should be more common. And that could be one mechanism by which biotic interactions influence the latitudinal gradient. And also the geographic mosaic is just this idea that you may have more selection going on because you're interacting with your enemies, and they are also evolving at the same time. So you have this sort of speed of adaptation might be faster in the tropics where most selection is done by biotic factors compared to the temperate zone where you have a lot of selection done by abiotic factors. And things like cold are not evolving in response to frost tolerance. That's Doug Shemsky's pet idea. All right. What about extinction rates? This idea of finer niches, narrow niches in the tropics, in recent reviews hasn't received a lot of support. A lot of the work by Bob Ricklefs and Steve Hubble have shown there's actually quite a lot of functional redundancy in tropical communities. However, if you think about the enemy-free space, this idea of the Jansen-Connell hypothesis and density dependence, most studies have shown high support for that. And the idea being that rare species get an advantage in these kinds of communities that are being regulated by natural enemy attack. And so if you think about that over larger scales, you could imagine that extinction rates could be reduced in areas where you had this enemy-free space being really important, and the Jansen-Connell was operating very strongly. And there's been very few studies that have compared this Jansen-Connell effect across latitudes. And so, usually when they do, they don't use the same methodology. So really what we need for biotic interaction studies is to study biotic interactions across large latitudinal extents that use the same methods, quantify biotic interactions, and the strength of selection at the same time. Okay. So, to conclude, I think that the time-integrated area / tropical conservatism plus climate stability hypotheses, they complement each other nicely. They have a very strong theoretical basis, and that I think that these drivers set this foundation for latitudinal diversity gradients. Bioregions that are old, have a lot of area, and low climatic stability have larger population sizes that are going to have higher extinction, higher speciation probabilities, and lower extinction probabilities. Energy itself, it definitely influences latitudinal diversity gradient, but indirectly, not directly. And it does so through population size, food for higher trophic interactions, and possibly through seasonality and generation time. Biotic interactions also probably play a role, but we need further study. So I've shown you this enough. I like this framework. This is just showing how energy may indirectly influence that framework. I don't think I have time to talk about this, but this is our paper with Walter, where it's shown that even though that energy actually plays a really good, is a very good predictor of understanding species diversity at the grid scale of 100 kilometers. So not the bioregional scale, but how species may be coexisting in more local scales. And so if there's time, we can talk about that later. But the idea being that there's a hierarchy to these predictor variables. How would we implement this framework? And basically, what you would do is choose your clade of interest, and this is going to be a big clade, like birds, not penguins, okay? And then you really have to draw a map on the world. What are the bioregions that are relevant to your clade of interest? And then you have to quantify the relevant window of time. You can do that with molecular phylogeny. You can say, how old is this crown group? And you know, how back and far in time do we want to go to be able to account for 70 or 80% or 90% of the extant species, or extant plus extinct species if you have a good group. And then you can correlate all these variables: time integrated area, climate stability, environmental heterogeneity, productivity, temperature, with either extant diversity, or you can use extant plus fossil diversity, or use phylogenies. I think paleoclimatic modeling is going to become more and more tractable. This is a paper of Rory's that he shared with me. This is not just going back to the last glacial maximum, it's going back 120,000 years. And this blue area is basically climate refuge. So you can imagine using this blue, and this is what Rory's doing, and thinking about, in this bird species, what's the genetic diversity in this climate refuge? And how does that correlate with climate stability? But you can imagine this being an approach to go together with those other variables I was talking about. So I'd like to thank funding sources, my seminar that I taught last year on this subject, Gary Mittelbach, Jim Valentine, Rory, Walter, and all of you for listening. I'd be happy to take any questions.
Go ahead, Paul.
So Paul, I'm thinking about one thing that you brought up here. Made me think about microbes. I'm wondering how microbes fit into the habitat. Some of them are symbiotic, some of them are parasitic. Do you see them being able to fit into these organisms in the same way? Yes and no. Drawing a bioregion that would be relevant for microbes is really tough, right?
Paul, can you repeat the question?
Oh, he asked, what about microbes? Can you imagine this framework being able to explain, say, microbial diversity? And my answer to that would be, it'd be really difficult for organisms that disperse as well as microbes to think about what would be the relevant parameters. How would you think about an evolutionary arena? And it might be that because microbes are such habitat specialists, that you might think about it in terms of habitats, not geographic regions. And maybe there would be a way for it, but your framework has got to be relevant to the biology of the clade of interest for sure.
So I had a thought about the speed question, because it bears on something I've been reading about. And there was this review on quantitative trait loci that in plants that are either underlying interactions between the plant and in the abiotic environment, or those in the biotic environment. And there's this, it feeds into Fisher's geometric model, but the idea is the same, that the adaptive peaks are other taxa, or other individuals in the case of a plant-herbivore interaction. And they found in this review, and it was sort of based on mostly model and crop species, so keep that in mind, but that overall the QTL were of larger effect size if they were underpinning a trait that was interacting with another organism.
Oh, cool.
Yeah. So it kind of feeds into
Yeah.
this macro evolutionary or sort of community level pattern. But I don't think anybody's really bridged that divide.
Great. Yeah, well, if you could package that off to Doug Shemsky, I'm sure he'd give you whatever you wanted.
And then what about the mutation limitation idea? Like, so are most species mutation limited in terms of adaptation?
Anyone know the answer to that?
No.
Right. I was waiting for Mike to say that.
No, I mean, I don't think we know the
Yeah.
answer to
It seems like in island radiations, you're starting out with very little diversity and you get all kinds of crazy things
Exactly.
happening pretty fast.
Yeah.
that. Well, there's there's the famous statement made by Dick Lewington in 1974 that there's no aspect, and this is in Drosophila, which has a big population size, but there's no aspect of the phenotype of Drosophila that you can select on and not get a response, suggesting that there's a lot of genetic variation for basically everything, and adaptive evolution is not mutation limited. But that's Drosophila, when I asked her.
So one of the best studied latitudinal gradients have been the great American interchange. And the thing about that interchange is that it includes many different time scales and many different lineages that have evolved over very highly variable time intervals. So I wondered if there is a way to impose a kind of an error bars on your hypotheses so that you get a sense of how reliable they are when you have lineages that are so varied in their relevant time frame and geographies that are so variable with respect to
But
how
I would
much
say
we know about
that they wouldn't, that they would be perfect, because what you have in the biotic interchange is you have lineages entering new evolutionary arenas and speciating over a relevant window of time. You have a few exceptions of things like mountain lion that just seem to come in and then not speciate. But for like rodents, for example, like they totally take off when they get to the neotropics. And that would just be part of I think that would be part of the reason why you get a latitudinal gradient, because they're doing exactly what we predict them to do. And the challenge would be to make sure you had the right window of time.
So how about the error bar idea of how reliable is a hypothesis under these sets of conditions?
Yeah, I don't know. I mean, yeah. I mean, any approach of taking the bioregional scale, you're going to get you'll get some error bars, right?
Yeah.
And that's part of those are your replicates that you use to test the hypothesis. So, am I not understanding your question?
No, not. I think I agree that the Central American interchange is a really great test of the different patterns that you're observing. It would be interesting to know what proportion of those patterns fall into these different categories.
I'm interested in this idea that the time area integral is missing the shape of the area over time curve, and wondering if you could expand, how would you incorporate the shape of the curve into the time area estimate to...
Well, the very simplest way would just be using the minima and just connecting dots through the minima over time.
Yeah.
That'd be the simplest way. But that might not be the best way.
So how associated are, or is total time area, the integral, with shape? Is there a big relationship there, a strong relationship, or?
So when we just did those shapes of time integrated area with tree diversity, we got significant correlations. I can't remember what the explained variance was. Probably not. Wasn't that great. I was probably 50 or 60% or something. Was that what you're asking? No.
Or I'm wondering if regions with a large time area integral also tend to be very stable, or
Climatically stable?
Yeah.
No, not necessarily.
Okay.
There are some large old temperate regions like that are much less climatically stable than large old tropical regions. But again, our sample size is low across the world with biomes and bioregions, so your statistical power is a little low.
Yeah, I actually have a follow-up to that. So is it, I know you're interested in the regional scale, but is there benefit to sort of parsing this in a finer way to give yourself more statistical power, or is that just missing the whole point?
I think it's missing the point, because what you're doing is sort of pseudo-replicating. You start double counting species when you're not using independent evolutionary arenas. You got to think about the scale at which speciation and extinction occur. Right. And unfortunately, that's a pretty large scale for most groups, not all, but most groups of organisms.
Well, I was curious about population size. So is there, and how it might relate to speciation and extinction rates, is there good evidence for a given biome where you can compare clades that are, say, sisters, where you think there have been differences in long-term effective population size on speciation rates and extinction rates? Is there good evidence that larger populations undergo more speciation and less extinction? The extinction is a little
Extinction
easier.
is pretty well documented.
Sure. What about
Speciation is tough because of how population size interacts with geographic range size. And it's kind of weird, like, when you have small geographic range sizes, then yeah, you have a low chance of running into a barrier, right? But if you have a really, really huge population range size, you might go around the whole barrier. And so some of those theoretical papers say that you would predict intermediate geographic range sizes to have the highest speciation rates. But I don't know if anyone's ever taken empirical data, and that would be really fun to do actually, and correlate population size with, you know, say diversification, speciation rate. And that would be a really cool way to test some of these ideas. And do you know of anything like that, Rory?
No, not specifically.
Okay.
I also was curious
Different
just about
question
the,
for
I
you.
mean, the implied relationship between geographic range size and population size. I mean, I certainly can think of lots and lots of counterexamples. Mostly I'm thinking of mammals, but you know, where there are things that have small effective population. Mountain lions are a good example. The effective population size for mountain lions is small. They have an enormous range.
But yes. You mean, you have to keep things equal, right? I mean, if you were comparing a mountain lion to a close relative of a mountain lion that had a similar ecology and size, right? I mean, I see your point, but
Well, that's why I was saying initially, if you had sister clade comparisons
Yes.
where you thought
Yes.
that the sisters had different effective population
that would be
sizes,
really
you could
cool. That's a great idea.
speciation rates through time.
I like that idea.
Well, some of us would argue that speciation rates are the same in the temperate environments as in tropical environments, and all that really matters is persistence. In other words, the extinction filter.
Yeah. I mean, you can make that like. There's definitely this concept of ephemeral speciation where, so Dolph Schluter has been writing about how, you know, you get high speciation rates at high latitudes, even higher. But I would argue that, but because the persistence is so much more important, that even though you may have high speciation rates, it's kind of evolutionarily insignificant. Because sticklebacks aren't very diverse, really.
But it's
And actually, he's never compared. I'd like to see a comparison of some sort of fish in a tropical lake, if you want to.
But if you look at birds, that pattern's played out, I mean, pretty well. That it might even be higher in temperate environments, but it all comes down to whether those lineages persist through time or not. So it's a misnomer. Sometimes people get focused too much on the speciation and they forget about the other side of the equation.
No, I definitely agree.
All right. Thank you very much, Paul.
Thank you.